Immune responses are not always protective in nature, but can be associated with tissue destruction and disease. It's now evidence that pathological immune processes in autoimmune disease use immune cells and mediators similar to those in protective immune reaction. Fundamental properties of the normal immune response, as we know: antigen-specific recognition, diversity of the lymphocyte repertoire, discrimination of self versus non-self, immunological memory, self-limitation. The ability of T cells to respond to an antigen depends on the availability of the correct antigen receptors. It's estimated that about a possible TCR are available in our bodies. This level of diversity can be generated by combination of different mechanisms such as multiple germinal theory, exon rearrangement or combination diversity, junctional diversity and junctional flexibility. These gene segments are often translated in all three reading frames: alpha and beta chain association. These TCR, beta and alpha chain gene recombination and expression, the germline DNA, rearranged DNA, and the primary DNA transcript, the messenger RNA, and nascent polypeptide TCR chain assembled TCR molecule. Tolerance, by definition, specific controlled inability to respond to antigen to which an individual has the potential for response, elimination or neutralization of the autoreactive B and T cells. A breakdown of this working system can cause autoimmunity. Physiologically, it's directed against antigen from the normal tissue, but it can be induced against any antigen by the use of appropriate procedures such as administration of antigen to very immature animals, administration of very large or very small dose of antigens, initiation of antigen treated in such a way that it could not be processed properly. When a specific lymphocyte encounter antigen, three possible outcomes may occur. The lymphocytes are activated, leading to immune response. The lymphocytes are inactivated or eliminated, leading to tolerance. The antigen is ignored. The choice between lymphocyte activation, tolerance, or ignorance depends on the nature of the antigen-specific T lymphocytes, nature of inducing antigens, nature of presentation to immune system, activation of naive T cell in the presence or absence of co-stimulatory signals, an overview of the lymphocyte response, the viral resistance, infected cells, cytotoxic T cells, and this is the macrophage and the mesohistochondrial presenting T helper cells, virus, interferon, interleukin, interferon gamma, interferon 2. This is the infected cell and the virus. Follicular dendritic cell uptake the microbe of microbial component. We discuss this in detail. Here the normal immune response, immunogenic antigen, proliferation and differentiation. Here the normal immune response, the tolerance, tolerogenic antigen, energy, functional unresponsiveness or apoptosis, cell death, immunogenic antigen, no response, ignorance, non-immunogenic antigen, immunogenic antigen. This is the fate of lymphocyte after encountered with antigens. The importance of tolerance, self-antigen normally induced tolerance. If we know how to induce tolerance in lymphocytes specific for a particular antigen, we may be able to use this knowledge to prevent or treat unwanted immune response. Self-tolerance phenomena. It's a tolerance to self-antigen, which is characterized by it's not inherited, it's antigen specific as in immune recognition. It's achieved through clonal deletion of the self-reactive lymphocyte through clonal energy of potentially autoreactive T and B cells. It may be overcome, leading to autoaggressive immune recognition. General feature of tolerance: dose-dependent, large or small dose of antigen are potent inducer of the tolerance. Site-dependent antigens ingested orally are more likely to induce tolerance. Delivery-dependent antigen in the absence of co-signal result in energy. Mechanisms of tolerance: central tolerance, immune self-reactive lymphocyte, self-antigen. Peripheral tolerance, mature self-reactive lymphocyte self-antigen. Some self-antigen are ignored by immune system. Mechanism of tolerance induction in T cells. Central tolerance, positive selection, negative selection. Peripheral tolerance, regulation, energy and inhibition, deletion, activation, induced cell death, immune suppression, ignorance, immune paralysis. Central tolerance, central mechanism of the induction of tolerance. Step CD80, CD24, antigen presenting cell, and here this signal and apoptosis occur, or another pathway, maturation and surviving of the cell and leave the thymus. The central mechanism of induction of tolerance. Positive selection, it's the selection of T cell carrying receptor which can recognize the self MHC antigen. This process occurs in the cortex of the thymus. If the thymocyte recognize self MHC with intermediate affinity, they will be stimulated to proliferate, leading to colonal expansion. If thymic epithelial cell present antigen in association with MHC class 2, the T cell will lose CD8, change to single positive CD4 T cells, but if they recognize MHC class 1, they will be signal positive CD8. The positive and the negative selection in thymus, in the thymus gland. This is occur inside the thymus gland. Negative selection, T cells with receptor binding with high affinity to the self-MHC self-peptide complex will be eliminated either by clonal deletion through induction of apoptosis, clonal energy by inhibition of interleukin 2 production. Negative selection will ensure the removal of self-reactive T cell, leaving only two T cells which are self-tolerant, MHC restricted, reactive to foreign antigens. Normal response and clonal energy, T cell energy, and antigen presented by co-stimulatory expanding antigen presenting cells. A normal cell response, if the T cell recognize antigen without co-stimulatory or in the presence of CTA, E4 B7 interaction, the T cell fail to respond and rendered incapable of responding even if the antigen is subsequently represented by co-stimulatory expressing antigen presenting cells. The normal response, antigen recognition. As we know, antigen recognition will with co-stimulation, the T cell response, T cell proliferate and differentiate. Co-stimulatory deficient antigen presenting cell restimulation with antigen presenting cell expressing co-stimulation, co-stimulator T cell energy. The T cell proliferate and differentiate. Here in this pathway, altered peptide ligand and failure to respond to native peptide. Peripheral tolerance. There are two signals required of T cell activation. The first signal is from the interaction between TCR and the peptide antigen. The second signal is from the interaction of co-stimulatory molecule CD28 T cell and the CD B7. The relation between CD28 and CD40. Mechanism of peripheral T cell tolerance. Ignorance, deletion, regulation, energy and inhibition, suppression and deviation, immune paralysis. Ignorance. Self-reactive lymphocyte may fail to recognize or respond to some self-antigen in the periphery, but the cell neither die nor become anergic. Mechanism of ignorance may be the level of antigen may be below the threshold required to induce the activation or deletion of T cells. The antigen may be physically separated from T cells. The antigen may be presented in the absence of co-stimulation, absence of T cell help, deletion, repeated activation of mature T lymphocyte by self-antigen, recognition of self-antigen without second signals, trigger pathways of apoptosis that result in elimination of the self-reactive lymphocyte. This process called activation-induced cell death. Mechanism of activation-induced cell death. Co-expression of the death receptor called FAS, CD95 and its ligand FAS ligand. Lack or an excess of the growth factor, especially interleukin-2, may stimulate or suppress immune response. Antigen recognition by T cell induces the production of pro-apoptotic protein, which normally antagonized by anti-apoptotic protein produced during immune response produced by microbe and not produced in case of self-antigen recognition. The normal response, antigen coagulation, recognition, T cell response, activation induced death on T lymphocyte. Here apoptosis, expression of the FAS and FAS ligand and this the apoptosis. Regulation, energy, inhibition. Energy, T cells that don't reproduce interleukin-2 on encountering their antigen and that therefore can't be completely activated are called anergic. Anergy may have widespread consequence because certain anergic T cells produce interleukin-10 which suppress the activation of T cells. No specific phenomic marker of anergy have yet been discovered. Inhibition, CD152, also referred to as cytotoxic T lymphocyte associated protein on T cells bind CD8 and CD86 on B cells with higher affinity than the co-stimulatory receptor CD28. By this way, CD152 inhibit the activation of T cells. The function of CD28, B7, B72, and CTLA molecules. The interaction between these here inhibition, here unresponsiveness, co-stimulation, the enzyme presenting cell, the T cell, the antigen presenting cell, and the T cell, and CD28. Cell separation. Some self-reaction to T lymphocyte may develop into regulatory cell whose function is to prevent and suppress the activation of other potential harmful self-reactive lymphocytes. Regulation of T cell may develop in thymus or in the peripheral lymphoid organ. Most regulatory T lymphocytes are CD4 and express high level of CD25. The feature of antigen-derived T cell to be suppressor and not effector, and the mechanism by which T cell inhibit immune response are slightly known. However, there are several ways in which T cell may suppress the function of the another cells. Soluble inhibitory factors, the civil servant model, soluble inhibitory factor, cytotoxic separation, idiotype mediated separation, inhibition of T cell activation, inhibition of the T cell effector function, normal response and his suppression. Here the normal response, naive T cells activated. Here the interferon gamma activated macrophage, interleukin 10 inhibit function of the antigen presenting gene. Here suppression is the process of suppression. Immune paralysis. It's considered a type of clonal energy resulting from a very high dose of antigen which bypass antigen presenting cells, reaches the helper T cell directly, making them energetic. Character of T cell tolerance: need low dose of tolerizing antigen, persistence of tolerant state of long term. It may be central or peripheral B cell tolerance. Character needs a high dose of antigen, persist for short time, can be induced either in bone marrow or periphery. Mechanism may be by deletion or energy. Inappropriate antigen presentation will lead to failure of CD4 to deliver help to B lymphocyte. Leading mechanism: several mechanisms available to filter autoreactive B cell out of B cell repertoire. Clonal deletion of immune B cell in the bone marrow, central tolerance, receptor editing, peripheral tolerance, clonal exhaustion, blockage of PCR, lack of help from T cells, clonal energy functional inactivation, the deletion of autoreactive B cell in the T cell zone of the spleen or lymph node, central tolerance negative selection, the immature B cell, apoptosis, mature B cell, receptor editing, expression of new antigen receptor, failure of central tolerance of B cells, and maturation of the B cell to peripheral lymphocyte, negative selection and receptor editing in immature B lymphocyte. The mature B lymphocyte, energy block in antigen receptor induced signal, exclusion of the B cell from the lymphoid follicle. Clonal exhaustion, repeated antigenic stimulation of B cells will convert the B cell into plasma cell. If all B cells are differentiated into plasma cell, no memory cell will be available for secondary immune response, leading to tolerance. Blockage of BCR. Some polymorphic antigens such as pneumococcal polysaccharide can bind irreversibly to B cell receptor and block any further response by these cells. Duration of tolerance dependent, resistance of the tolerizing antigen, the ability of bone marrow to generate fresh antigen sensitive cells, and the factors of the favor simulation of the immune response factor that favor tolerance, the amount, the persistence, portal of entry, presence of adjuvant, properties of the antigen presenting cells. Here the amount, optimal doses, here high dose, fever tolerance, persistence, prolonged, repeated, short-lived, port of entry, subcutaneous, intravenous or oral, presence of adjuvant, antigen with adjuvant stimulate helper T cell, antigen without adjuvant, non-immunogenic, properties of the antigen presenting cell, high level of co-stimulatory, here low level of co-stimulatory, and cytokines. Self-tolerance in T and B lymphocyte. This difference, principle of site tolerance, T lymphocyte and B lymphocyte. Principal site of tolerance, thymus and periphery, bone marrow and periphery. Tolerance sensitive stage of maturation, double positive thymocytes, immature IgM, IgT. Stimuli for tolerance induction, central high affinity recognition of the antigen, peripheral lack of co-stimulatory or repeated stimulation, central in B cells recognition of the multivalent antigen in bone marrow, peripheral antigen recognition without T cell helper. Principal mechanism of tolerance, central clonal deletion, peripheral energy activation induced cell death and suppression. Central in B, clonal deletion, peripheral block in signal transduction and failure to enter lymphoid follicle. Definition: it's an immune response against the self-antigens and is important cause of death. Autoimmunity is an immune response against self-antigen and is important cause of death. 5 to 7% of adults in Europe, North America, two-thirds of are females. More than 40 human diseases are of autoimmune origin, such as SLE, RA, and IDDM. A small number of B cells which reactive to self-antigen are always present in the lymphoid organs. If regulation of these autoreactive cells break down clones of lymphocyte can generate high level of autoantibodies or autoreactive cell leading to autoimmune damage. Spectrum of autoimmune response. Autoimmune potential present in every body reflect T and B receptor diversity, healthy response. Physiological autoimmunity, sporadic, related to transient tissue damage, may have a homeostatic rule. Pathological autoimmunity, rare, result of complex interaction of genetic and environmental factor. Categories: organ-specific, the autoimmune reaction are directed against one or more specific cytoplasmic constituent, plasma membrane structure, or secreted product of the cell. Non-organ specific, the autoimmune reaction are directed against a structure common to many tissue and found through the body, like nuclear component, mitochondrial proteins, or constituent of muscle. Here we found a table, myasthenia gravis, the spectrum of autoimmune disease, the disease, and the maximum principle of autoimmunity. Normal or physiological autoimmunity. More than one abnormality are required to induce autoimmune disease. A heterogeneity of tissue destructive mechanism, diversity of initiation agents, the multifactorial pathogenesis of the autoimmunity. Normal or physiological autoimmunity. It's not true that all autoimmune responses are bad cause diseases. Some autoimmune responses have important physiological function, are essential for the normal functioning of the body. Example of normal autoimmunity: the action between CD4 and CD8 T cells and the measure histocompatibility class 2 and or class 1 respectively. Anti-idiotype antibodies: the antibody binding site idiotype on immunoglobulin molecule can function as epitope, provoke production of antibodies. Anti-idiotype: each anti-idiotype response tends to exert a negative feedback on the preceding response. Therefore, the net effect is termination of antibody response. Cell-cell interaction mediated by adhesion molecule and their ligands. Removal of the aged RBCs. As RBCs become aged, an onion transport glycoprotein called band 3 will be cleaved. A new epitope will be formed, which will be recognized as autoantigen. Interaction between these antigen antibodies will facilitate phagocytosis of RBCs. The band 3 protein is also present on platelet, lymphocyte, polymorph, and renal cell. More than one abnormality is required to induce autoimmune disease. It's more likely that more than one mechanism is impatient is dysregulated before an immune response acquires pathological significance. Heterogeneity of tissue destructive mechanism. Multiple different cells types such as antispecific lymphocyte, non-specific phagocyte, and granulocyte fibroblast infiltration autoimmune inflammatory lesions. Several agents can initiate an immune response in the microenvironment of the affected tissue. The genetic background of the patient finally lead to pathology. The multifactorial pathogenesis of autoimmunity. Autoimmune diseases develop in hosts with multiple inherited risk factors combined with environmental contributions. Factor affecting severity of autoimmunity. Quantity factor, how much autoantibodies present, how great the T cell response, how much of the cytokine produced. Quality factor, antibody affinity, isotype, fine specificity. Genetic factor, either augmenting or allowing susceptibility. Environmental factor, infection, chemical, food, UV. Induction of autoimmunity. Many autoimmune diseases show spontaneous remission, exacerbation, suggesting an stable relationship between positive and negative and suppressive cytokines. Initial triggering, formation of autoreactive cells, T cells, normal pathway, special circumstances, gene, complement, hormone, negative selection, no problem, failure to negative selection, autoreactive T cell or autoantibodies, tissue damage, and autoimmunity, and autoimmune disease, and autoimmune patient. Here, the generative lymphoid organ, abnormal selection or response, self-reactive lymphocyte, cytokine, other stimulus, presentation of self-antigen to autoreactive lymphocyte, tissue injury, and autoimmune disease. Theories of autoimmunity, failure of thymic education, breakdown of peripheral tolerance, antigen non-specific lymphocyte activation, molecular mimicry, abnormalities in lymphocyte interaction, hormonal factors, handedness, environmental factor, exposure to of hidden antigen, failure of thymic education, removal of self-reactive lymphocyte from T cell repertoire is achieved during thymic selection through clonal cell deletion or energy. Escape of autoreactive T cell from the thymus could supply harmful T cell to peripheral lymphoid tissue, rheumatoid arthritis. Breakdown of peripheral tolerance. Peripheral tolerance is considered as a security system to prevent activation of escaped self-reactive T cells. Failure of peripheral tolerance may be due to apparent expression of co-stimulatory in tissue, antigen presenting, or mutation of CTLA4 may result in breakdown of T cell energy and the tissue-specific autoimmune reaction. Mutations that interfere with the apoptotic death of mature lymphocyte may result in autoimmune diseases such as mutation in Fas and Fas ligand. 3. Defect in T cell mediated suppression may contribute to autoimmunity. Self-reactive lymphocyte inflammation, innate immunity, inflammation. Here, influx of self-reactive lymphocyte into tissue, activation of autoreactive lymphocyte, tissue injury, autoimmune disease. Antigen non-specific lymphocyte activation, systemic autoimmune disease such as SLE are characterized by the production of a wide array of autoantibodies, suggesting the activation of polyclonal T lymphocytes. Molecular mimicry, sharing or structure similarity between epitopes of infectious agent and the tissue of the host. Initiated immune response leading to formation of antibodies or primate T cell against these antigens. The initiated immune response will be directed against normal host tissue causing autoimmune damage. The peripheral immune repertoire or molecular mimicry, endogenous host determinant, the memory T cell defenses, and autoreactive T cells. The mechanism by which microbe may promote autoimmunity and self-tolerance. Here, antigen presenting cells, the resting tissue, antigen presenting cell, and T cells. Self-tolerance energy, this is the self-tolerance, induction of co-stimulatory stimulators on antigen presenting cells, microbe activation of ABC, self-reactive T cell autoimmunity, molecular mimicry, microbial antigen, self-reactive T cell that recognize microbial peptide, activation of T cells, self-tissue autoimmunity. Examples: rheumatic fever, sharing is between the cell wall of M protein on the group Staphylococci, the cardiac myosin. Acute glomerulonephritis, sharing between certain strains of B hemolytic streptococci and the basement membrane of the renal glomeruli. Multiple sclerosis, sharing between Epstein-Barr virus DNA polymerase and myelin basic protein. Myasthenia gravis, sharing between poliovirus capsid protein and acetylcholine receptor in motor in plate and tissues. Ankylosing spondylitis, sharing between HLA-B727 and bacterial antigens such as Klebsiella pneumoniae. Abnormalities in lymphocyte interactions. Most autoimmune disorders developed as a result of emergence of T and B cell that had previously suppressed by the normal control mechanism of body. Th1 cytokine downregulate Th2, the reverse true. Rheumatoid disease are four-fold higher in lymphoid malignancy due to lymphoid tumor may result from a failure of immunological control mechanism with failure to self-tolerance. Some tumor represent the development of forbidden clone of cell producing autoantibodies. Lymphoid tumor may rise from chronic stimulation of immune system by autoantigen. Exposure of hidden antigen. Some human antigens are present in areas not visited by immune cells, such as testicle, CNS. Therefore, lymphocytes may not be completely tolerant to them. Entrance of these antigens into circulating following trauma will stimulate immune system, leading autoantibodies or cell against them. Hormonal factor. Autoimmune disorders are more common, more severe in female than males, which may be due to hormonal difference between them. Estrogen stimulate enhancer element of interferon gamma genes, which increase production of interferon gamma. Interferon alpha upregulate interferon gamma. Interferon upregulate the expression of MHC. Co-stimulatory molecule activate macrophage, display a major role in pathogenesis of tissue damage. Interferon gamma. Left-handedness and autoimmunity. Left-handed individuals are more directed to develop autoimmune disease than right-handed. One study shows that 11% of left-handed, 4% of right-handed suffer from autoimmune disease, mainly thyroiditis, ulcerative colitis, celiac disease. It has been suggested that left-handedness and immune malfunction may both result from abnormal endocrine function in fetal life. Environmental factor. Environmental factor. Virus as inducer of autoimmunity. It has been suggested that virus, especially those infecting lymphoid tissue, may interfere with immunological control mechanisms. The ones of IDDM in children, Coxsackie virus, the presence of Epstein-Barr virus, EB virus genome in salivary gland, or Sjogren's syndrome patient, presence of type C retrovirus or paramyxovirus in SLE, genetic susceptibility for autoimmunity. Family and genetic studies indicate that many genes affect the development of autoimmunity. Evidence the relative of patients with Hashimoto's disease have anti-thyroid antibodies may develop thyrotoxicosis or other autoimmune diseases. Studies with identical twins show that when one has multiple sclerosis, the other has a 30% chance of developing the disease. genetic risk factor, autoimmune disease, immune response gene, polymorphic HLA sequence, complement gene, T cell receptor, immunoglobulin gene, cytokines genes, and gene controlling antigen processing presentation, non-immune genes, mutation in any of the gene regulating programmed cell death, gene regulating response to tissue injury, gene regulating tissue repair, a defect in cytokines production or signaling that can lead to autoimmunity, the cytokine or the problem, the defect and the result, the factors that influence the pathogenesis of T-cell mediated autoimmune diseases, genetic susceptibility, activation of the autoreactive T-cell antigen-specific antigen, non-specific infiltration of the target cells, damage to the target organ. HLA disease association, it's now firmly established that HLA gene influence susceptibility for large group of diseases such as RA, HLA-DR14, MS, and HLA-DR2, SLE. Hypothesis of HLA disease association. Hypothesis 1. Peptide selection model suggests that HLA allele are essential molecule for binding of initiation antigenic peptides, which can induce a virus T-cell response combined with tissue injury or would fail to stimulate T-cell response. Hypothesis 2. Molecular mimicry model suggests a sharing between HLA structure and infectious agent. Hypothesis 3 suggests that TCR are responsible for autoimmune. The HLA allele associated with disease is due to its association with TCR. Gene is linking disequilibria rather than HLA gene. Are disease relevant such as cytokine and transporter genes. Mechanism of immune-mediated tissue destruction. Type 1 immediate hypersensitivity. IgE bind to the mast cell and when exposed to antigen they promoted released mediator facilitating the clinical manifestation of the allergy. Type 2 antibody-mediated disease. Autoantibodies with complement or cytotoxic T cell can cause lysis of the cell such as autoimmune hemolytic anemia, autoimmune thyroiditis, immune thrombocytopenia, myasthenia gravis. Complexes formed by specific antibodies and the antigen combined deposited into tissue and cause an intense inflammatory reaction such as SLE and vasculitis. Type 4 T-cell mediated. Many lesions in the autoimmune disease are heavily infiltrated with mononuclear cells. Autosensitized T lymphocytes contribute to pathogenesis of disease such as IDDM, ulcerative colitis, MSE, thyroiditis. Therapeutic implication. Therapies are needed that not only prevent T-cell reactivity but also establish tolerance once the autoreactive T cells have been activated and an autoimmune disease is diagnosed. One example of immunomodulation is the treatment of patients with hemophilia. In about 30% of the patients with hemophilia A who receive infusion of factor AA8 concentration, systemic injection of deaggregated antigen induce tolerance in helper T cells and can prevent experimentally induced autoimmune encephalitis. A peptide that inhibits the activation of T cell clone is called an antagonist. Some viruses escape the host immune response by producing such antagonistic variant of important antigen, and the administration of antagonist peptide has prevented autoimmune encephalitis in mice. Immune modulation. Because treatment with antibody against molecule important for the T cell function induced the T cells to become unresponsive in rodent. Several of these antibodies have been evaluated in clinical studies. A subform of CD152 inhibit the interaction of the co-stimulatory molecules CD8, CD86 on antigen-presenting cell with their shared receptor CD28 on T-cell treatment with this form of CD152 prevent or ameliorated lupus, autoimmune encephalitis, and arthritis in mice. T-cell can be categorized according to the cytokine they produce. Type 1 helper cells produce mainly interferon, tumor necrosis factor, and the interleukin 2, where type 2 helper T2 cell produce mainly interleukin 4, interleukin 5, and interleukin 13. Since multiple sclerosis and type 1 diabetes are mainly mediated by T1 helper cells and allergic disease by T2 helper cell, alternation of the cytokines balance is an appealing therapeutic possibility. Clinical trials that are evaluating the immunoregulatory cytokine interleukin 10 with chronic inflammatory disease have reported encouraging initial results in patients with psoriasis. Antagonizing the inflammatory cytokine tumor necrosis factor with monoclonal antibody or soluble decoy receptor have proved remarkably effective in treatment of rheumatoid arthritis and Crohn's disease. Purging autoreactive T cell from the T cell repertoire. Purging autoreactive cells from the autoimmune repertoire is an option in several cases of autoimmunity. Autologous stem cell transplantation has been tried in patients with rheumatoid arthritis, psoriasis, and systemic lupus erythematosus with variable success. It's not yet possible to judge the long-term efficacy and safety of this approach, and it's unclear where the patient who receive such treatment can generate completely new repertoire of the T cells. Nevertheless, purified CD34 hematopoietic progenitor cells that are depleted of autoreactive lymphocyte and inflammatory leukocyte may remind to reduce tolerance